And we are bringing together scientists which are interested in the interface between the nervous system, metabolism, organ physiology, immunology. So stay tuned for updates in our website and worldwide neuro seminars. So the talks this year will still use the Crowdcast platform. During the seminars, you can ask any questions using the Ask a Question button. And also, if you want, you can meet the speaker after the talk during a Zoom Q&A session. Unfortunately, today our speaker won't be able to be with us after the talk. As you can see, she's in a hotel room ready for giving wonderful talks to other people as well. So today I'm eager to introduce Dr. Staci Bilbo. She's currently a professor of psychology and neuroscience at Duke University. She received her bachelor's in psychology and biology from the University of Texas at Austin, and her PhD in neuroendocrinology at Johns Hopkins University. She was previously on the faculty of Duke University before she joined the faculty at Harvard in 2016, where she not only served as an associate professor at Harvard Medical School, but also she was the director of research for Lurie cancer for autism at Massachusetts General Hospital for Children. She returned back to Duke University in 2019 as the Holy Family Professor of Psychology and Neuroscience, but she still maintains her relationship with Harvard University to continue her research collaboration in Boston and beyond. So Dr. Bilbo's research is broadly focused in neuroimmune interaction during brain development, and particularly how microglial activity can affect both normal and abnormal brain development. Today, she will be talking about microglia, microbes, and neurodevelopmental disorders. So without further ado, the stage is all yours.
Thank you very much for the invitation. And hopefully you can now see this.
It is perfect. We can see it.
Perfect. Okay. Yes, thank you again for the invitation. This is obviously a really good fit with the research that we're interested in my lab. And so I'm excited to tell you about what we've been working on for the past few years, and including some very new data that we've just recently collected. So as was mentioned in the kind introduction, we're very interested in the role of innate immune signaling within the central nervous system and how this is important in both health and disease outcomes. And so I think, as many of you know in this audience, there's a lot of interest in microglia in recent years. And we know that they are not simply immune cells that sit in the brain and wait for something to happen, but they are indeed extremely dynamic cells and are continually surveying their environment, sampling synaptic activity, essentially listening to what neurons do. And we know that that can have a profound impact on the function of those neurons and therefore the brain. Of course, the question becomes, what are they doing? Why are they listening to neurons in the first place? And we know that they're doing lots of things, but just as macrophages in the periphery are good at eating things, which is pictured here, a macrophage about to engulf bacteria, we know that microglia are also very phagocytic cells and are good at eating things as well. And so in early brain development, one of their most critical functions, we now know based on the work of Beth Stevens and Dory Schaefer and many other labs now have shown there's this critical role for microglia in pruning synapses during critical windows of development, and that this happens in an experience-dependent fashion. So that's one reason we think they're listening so intently to neurons. Of course, the question for me when this literature really started to emerge was, again, the question of why. So why on earth do we need immune cells in our brains to do something like prune synapses when presumably the brain has mechanisms to do this all on its own? And it does. And so why would we need sort of a complementary mechanism that is quite literally part of the immune system? Because microglia are brain macrophages. Well, nothing in biology makes sense except in light of evolution. And I don't need to tell anyone right now in our world that we live in a dirty world. And I've been using this slide for many years, but I recently had to update it for our new companion. But the fact is that this is not new. This pandemic feels very new to all of us, but we've been through pandemics before, and throughout our entire history on Earth, people have had to try to fight off infections and other things that make us sick. So we're sort of in a continual arms race with pathogens and things that make us sick. So if we can argue that essentially one of the biggest tasks that we perform each day is simply surviving infections, not getting sick, we have to reason that we invest everything that we can into overcoming those infections or those pathogens. And it turns out that our arsenal includes changes in behavior. So if you think about the last time you were sick, which hopefully wasn't recent and isn't right now, but if you just think about it, it's easy to recall how you felt. And there are all of these conserved sort of behaviors and changes in motivation that occur that are actually an evolved adaptive part of our immune defense arsenal. And so we perform these behaviors as a part of our own host immune response. And so this rapid reorganization, of course, involves the brain. And this is one major reason that I believe we have so many immune cells in our brains, so that we can rapidly alter motivation in a way that is that's beneficial for our survival. And indeed, my lab showed a few years ago that microglia don't only prune synapses, but they very selectively prune dopamine receptors within the developing nucleus accumbens during this very precise window of adolescence. And that this pruning behavior of these receptors is really critical for the development of normal social behavior. And if you inhibit their ability to prune these receptors, then social behavior does not develop normally. So this sort of brings us back to what is really the $1 million question for the work in my lab, which is, if we know that microglia are positioned in the brain so that they could rapidly alter neural activity through synaptic pruning and other mechanisms as well, and they're important in health and important in healthy brain development, then what's the sort of progression from health to disease? Because we also know that they are implicated in many, many pathologies. And that's really the $1 million question. So for my lab, it's been really useful to think about how these two systems first develop. So sort of going back to the beginning. So very interested in how immune activation during critical neurodevelopmental windows may increase that risk of pathology, may sort of tip us over into disease. And this is based on a strong epidemiology, which links early inflammation to increased risk of a lot of pathologies, and not only neurodevelopmental disorders, which we work on quite a bit, but even neurodegenerative processes much later at the other end of the lifespan. And so broadly, this is what my lab is interested in. We're very interested in when these microglial precursor cells from the yolk sac come into the developing central nervous system, and when they're sort of progressing through their own critical windows of development, what happens if you have an immune activation? And we look at a diverse number of different environmental factors which could activate the immune system, potentially impact microglial development, and then what are the consequences for later life. And you'll note I have these sort of two trajectories here, and this is quite intentional because we know that there are sex differences in microglial development. And we're very interested in sex differences generally because all of these later life outcomes that we're interested in tend to have sex biases in their presentation. And this is a really understudied area. So this is an area that we care a lot about. So today I'd like to share with you some data from a model in which we're using a mouse model to look at autism-like behaviors, because we've become quite interested in this particular neurodevelopmental disorder in the last few years. If you're not so familiar with this, it is a pervasive neurodevelopmental disorder, and it's quite broad and heterogeneous in its presentation, but at its core, we have behavioral changes like social deficits and communication deficits, repetitive behaviors, and things like that. And it's one of the most sex biased. It's about 1-to-4 ratio, many more males compared to females, which is also interesting to us because there's very little known about why this is. So over the years, there have been many animal models that have been developed to try to get at this question of whether immune activation during development might occur in a way that could lead to autism. And many labs have worked on this over the years, which generally involves giving either an infection or an infectious mimic, like poly-IC, to a pregnant mouse dam, not a human mother, mouse or rat, and then looking at how the resulting cytokine production as a consequence of that infection impacts the fetal brain and therefore behavioral development. And so the vast majority of these models have really focused on infectious or microbial origins of this disorder, which again is well founded in the epidemiology. Now, that said, as I told you at the beginning of this talk, infections are not new. This is not a new thing, and we know that there are a much broader array of environmental factors that may be implicated in autism spectrum disorder, and sort of lots of things that could activate the immune system in a way that could alter brain development. And if we look at how autism prevalence has changed over the past several decades, as many of you probably know, it's been a really startling increase over time. Some of this is undoubtedly increasing diagnostic criteria, but we believe that some of it is a real increase. And again, infections have not really changed, maybe the past year and a half notwithstanding, but we haven't had any increase in infectious diseases. But at the same time, we've also had a striking increase in autoimmune disorders broadly. Again, suggesting that there are other environmental factors that are impacting our immune system. And from an immunological perspective, this makes a lot of sense. We know that the immune system doesn't simply respond to so-called pathogen-associated molecular patterns, which are present on bacteria and viruses and fungi, etc., but it responds broadly to danger signals, danger molecules, which are released as a consequence of cellular death or cellular distress, and that then broadens the sort of scope of what could be impacting our developing immune systems and therefore our brains. So the work I'll tell you about today briefly is work we've done in collaboration with the Environmental Protection Agency for several years now, in which I became quite interested in the role of air pollution as a toxicant, which could impact the way microglia respond based on some evidence that air pollutants might actually be activating innate immune cells in a very similar way to something like bacteria. And we now have a lot of evidence that this is the case. And it's also one of the strongest epidemiological links to autism in humans. Now, this said, even with something strongly linked to autism like air pollution, the associations of single factors tend to be rather weak. And we know that the literature tells us that it's typically multiple factors. So either a genetic vulnerability combined with an environmental factor, or in this case, what we became quite interested in is this literature, growing literature showing that psychosocial factors like maternal stress in the case of developmental disorders can increase vulnerability to an environmental toxicant. And so this is the model that we use in my lab to try to get at these questions. So first of all, we expose pregnant mouse dams intermittently to either a vehicle solution or a proxy for air pollution, diesel exhaust particles, which are one of the primary toxic components of the air pollution that we all breathe, depending on where we are in the world, of course. But this is environmentally relevant concentrations for Durham County, where I live. We do this intermittent exposure to kind of model what it would be like to live consistently in one environment. And what we know is that this causes a persistent but very low-grade inflammation within the lungs of the dams. And then to combine it with that second hit, that stressor component, which we predicted might be unmasking the effects of the diesel exposure, we do a nest restriction paradigm in which the moms have the bedding taken away and they're on this wire mesh. And then they have some of the nest material removed, which sounds terrible, I know, and it is stressful, but otherwise they're freely moving. They have all of the things that they need, food and water, but they simply have less nest material during that last third of pregnancy when they're quite literally in nesting mode, preparing for their pups. And the lab that developed this model has shown that this causes an increase in stress in the dams, and we see this quite strikingly as well. So in the case of the 2-hit group here, those exposed to both diesel particles and maternal stress, they have higher CORT levels during pregnancy compared to just the mock installations. What we don't see, however, is any change in maternal care because we restrict this stressor to the prenatal period, and before they have their pups, they have just normal housing conditions given back. And we've carefully measured maternal care, and the moms are caring for their pups just as well. So we don't have that sort of confound in interpretation. What we do see is a striking maternal immune activation, including several pro-inflammatory cytokines, which have actually been shown to be increased and to be causally implicated in the infection-based models of maternal immune activation. So it's a striking similarity and actually sort of confirms our initial hypothesis that these other environmental factors might converge on the immune system in a similar way. So we have maternal immune activation, we have a stress response in moms, and then of course the question is what's the impact on the pups? And in the interest of time, I'm just going to focus on the social behavior change here, but we do see communication deficits early in life in pups exposed to the double hit of stress and diesel exhaust as a proxy for communication, and this persists into adulthood. And when we look at social behavior, which is a sort of a classic sociability assay, and this work was done by Caroline Smith, a really talented postdoc in the lab, in which we simply ask, do they prefer another mouse or do they prefer an object? And here we see a really striking sex difference, which is consistent with autism, where only males exposed to the prenatal stressors have a decrease in sociability. So no preference between object and the mouse, whereas the females are unaffected. So the question becomes, of course, what are the mechanisms? And the final thing I'll tell you on this slide too is that if we just look at the impact of the stress alone or the pollution alone, we see no impact on behavior. So it really is a synergy and requires the two together. So what are the mechanisms there? And here, of course, we know there's a lot of heterogeneity in autism and a lot of mechanisms have been examined. But if you look at not the genetic mutations that are associated with autism, which accounts for that heritable component of autism, which is, of course, important. But if you look at what are the transcriptomic changes that you see, the gene expression changes that are consistent with an autistic brain versus a typically developing brain, there's been a very consistent pattern that has emerged. And that is that you see a downregulation of genes that are important in synaptic function and an upregulation of genes that are important for immune function. And this has been replicated in many large data sets at this point. So the first question we had was whether there are synaptic changes in this model. But that led us first to another question, which is where do we look? So as I've just said a couple of times, autism is heterogeneous. It involves many different brain regions, and to try to narrow in and look for a place where synapses might be altered was sort of a daunting task. So we decided to take an unbiased approach and collaborate with Kaf Sarasa at Duke, who does this really amazing multi-electrode recording sessions where they implant electrodes into multiple different brain regions, including amygdala, parts of prefrontal cortex, nucleus accumbens, hippocampus, thalamus, and VTA, all regions that have been implicated in social behavior before. And they simultaneously record from these brain regions as the mouse is interacting with either another mouse or an object. And they do this over repeated sessions, so they develop this huge data set. And then they use a machine learning approach to basically distill down what is the social circuit that's active during interactions with this mouse by asking, how active is a given brain region during this time, and then which brain region is talking to which other brain region, and what's the coherence of that communication. And the critical question is, could you look at the circuit activity on its own and predict whether our mouse is interacting with another mouse or not? If it's successfully predicting that, then they have successfully identified that social circuit. And that's exactly what they found. So if you look across all of the mice in our study, they found that the social network activity that they've identified is greater when the mouse is interacting with another mouse, which makes sense. But more importantly, we find that the amount of activity in this social network is greater or positively correlated with social preference. So in other words, the more they prefer the other mouse, the more activation of this social network you see. And this is the case for every group of mice that we had in our study, except for one, and that was our 2-hit males. So this is the control males where you see that strong correlation, and you see the same thing in both groups of females. But in our males exposed to the prenatal stressors, there's no correlation. So something is different about how their network is working. It is working, but it's not predicting behavior. So it's different, right? And this is just looking at what they call an electome, which is just mapping out the coherence of activity between different brain regions. Of course, our question then, as cell biologists, was to say, okay, so what part of the social network is broken so we can see if there might be synaptic changes there. And this is where Koff would always remind us that it's a circuit. So if you break any part of the circuit, it will be broken. But that said, one part of the circuit that was quite altered in our mice, which stood out to us, was this connection between the thalamus and the cingulate cortex. And so the anterior cingulate in particular has been implicated in autism. So many of the behaviors that are critical in autism or are critically disrupted in autism are mediated by this brain region. And there's a lot of literature showing changes in this brain region in autism, including hypoactivation and reduced connectivity between the thalamus and the ACC. So during early brain development, the thalamus sends these long-range excitatory projections into the synaptic layers of the ACC, and we can measure these synapses using a marker called vGlut2. So this is where we started. And this is work in collaboration with Çağla Eroglu, also at Duke, and a really fantastic PhD student that worked with both of us on this project. And what she used was an immunohistochemical approach to simply label for the pre- and postsynaptic markers and see where they co-locate. And based on some preliminary data that she collected, she knew that these synapses really begin to form at postnatal day 8, and then they begin to stabilize around P15. And then we looked later at P100 to see how persistent changes might be. So this is first looking at males at postnatal day 8. And what Karina found is a more than 2-fold increase in the number of these vGlut2 positive synapses in our 2-hit males, so males exposed to the double stressors. But by P15, this now reverses. So we now have fewer, and this persists to P100. So it's this early huge overshoot followed by an atrophy, which is really interesting. It's not the same level of analysis, but in many patients with autism, it's been described that there can be an early brain overgrowth followed by an atrophy. So it's sort of a similar pattern. And this is really striking because it's only in males. We don't see the same pattern in females at all, consistent with the circuit change and consistent with behavior. So are there changes in microglia? And the prediction being that because these cells are good at eating synapses, maybe they are impaired in their refinement of these synapses. And first, Karina confirmed that just normal wild-type mice, their microglia, are they actually eating these synapses early in development? And indeed they are. So Karina found that precisely between postnatal day 8 and postnatal day 10, microglia are just chock full of VGLUT2, which you can see in magenta, and CD68 is a lysosomal marker, which we know is important for the degradation of that material. So between P8 and P10, they're chock full of VGLUT2. So what happens to our mice? Is it altered in our combined stressor offspring? Well, one thing I didn't tell you about the previous pictures is that for these studies, Karina actually consistently used a couple of different immunohistochemical markers for microglia, one called P2R12, which labels all of the processes really nicely, and one called IBA1, which labels the cell body nicely. And this is simply to just get full coverage of the cell. And in the wild type mice, of course, both of these markers just stained every microglia. So it was a really nice way to do this. But what Karina immediately noticed when she did this in our experimental animals is that these two markers were now labeling distinct subsets of microglia, which we had never seen before. And you can see it much more clearly if you sort of look down a stripe of cortex, that there are patches of microglia that are exclusively labeling for only one marker. Again, quite striking. But it's actually sort of reminiscent of data from Beth Stevens' lab showing that there is a lot of heterogeneity in microglia early in development, especially prenatally. This really evens off during the first postnatal week. You don't see it in normal adult mice, but it can be reactivated with pathology. So the question for us was, are we actually seeing maybe an increase in this heterogeneity or a delay in the maturation of microglia in one group? And so that's exactly what Karina found. So if she measured the number of microglia that were co-labeling for both, which is sort of normal microglia, versus ones that are just exclusively showing one marker versus the other, she found that there's much more heterogeneity in our two hit males compared to controls. But again, this is gone by P25, so it's a pretty narrow developmental window. She also found that this is really primarily driven by one subtype, which are these cells that express P2Y12 really highly and do not express IBA1. Now, the dogma in the field of microglia is that these P2Y12 high cells are just surveillant homeostatic microglia. But we're wondering now, based on these data, whether this might be true. And in a moment, I'll show you that that doesn't seem to be true. So, but the take home here is that the diesel stress in males is increasing microglial heterogeneity. And importantly, it's not because there's just a change in the number of cells. They have the same number of cells between the two groups. So that's not describing it. The question is there a functional difference in these different subtypes? So first, Karina asked, is there a difference just in lysosomal content in the three flavors here? So these are just normal co-labeled cells, and these are IBA1 high, and then our P2R12 high, which seem to be the predominant type in the diesel stress mice. And indeed, the P2R12 high cells have half the amount of this lysosomal marker, and therefore they're engulfing fewer than half of the synapses. So they're impaired in their ability to clear these synapses. So putting this all together, we have a much decreased phagocytic index in our diesel stress males. So they're engulfing fewer synapses. So the critical question now becomes is this actually causally implicated in the social behavior changes that we see? So just to summarize everything I've shown you so far, what we've seen is that we have these two sort of very disparate environmental factors which have been implicated independently in autism, but neither alone very strongly. Now we've seen that they really synergize. They activate microglia. In data I haven't had time to show you, we know that this is actually dependent on the innate immune receptor TLR4, which is the same receptor that responds to bacteria. So this is an innate immune activation of microglia. This results in this really intriguing heterogeneity with a subtype with this impaired phagocytic capacity, this overshoot of VGLUT2, which then leads to an atrophy, and that mechanism we're still working on, and then ultimately social deficits and circuit deficits. Again, though, the critical question here is this causal, or is this all sort of correlative? So we wanted to show that definitively as possible. Of course, it's harder to rescue phagocytic function. So the approach that we took was, okay, well, what if we just take normal wild type mice and impair their phagocytic function? Do we see the same phenotype? So that's the approach that we took. And to do so, we used a molecule called neutrophil inhibitory factor, which actually binds very selectively to CD11B or CR3 receptor on microglia. And in previous studies in my lab, we've shown that this very precisely inhibits phagocytosis by microglia. And so we put this bilaterally directly into the ACC at P7. So it's right before they're needing to prune those receptors, the synapses rather. We put it in at P7, and then around P30, we once again did the sociability assay. And first, what we found is indeed NIF did knock down the amount of phagocytosis by microglia on P8. And when we look at sociability at P30, the NIF injected mice show no preference for the mouse over the object compared to the PBS injected controls. So the microglia loss of phagocytic function during this time basically phenocopies the synaptic and social deficits. So going back to this, the next question, of course, we have is how might we be able to rescue microglial function? So we have impaired function. And again, this was a quite surprising finding to us. There's a lot of interest in neuroinflammation and lots of disorders and overly exuberant microglia. Maybe they're pruning everything. Maybe they're overly inflammatory. We're not finding that. We're finding that microglia are impaired, and we need to be able to rescue their function in order for them to do their jobs properly. So that's the question that we're now considering, and we're coming at this from a number of different sort of directions. But one I'd like to share with you briefly here at the end is work we've been doing on the gut-brain axis. So going back out to the brain-body interaction, can we maybe look to other parts of the body to be able to impact the brain? And so there's a lot of interest in this, of course, as well in neuroscience broadly. So probably many of you know about this, but we know, of course, that the gut components, what's present in the gut, can profoundly impact the brain. The reason we specifically got interested in this is that gut dysfunction is often comorbid with neurodevelopmental disorders, including autism, up to 50% of individuals. We know that mice lacking microbiota, so germ-free mice, often display very robust social deficits, and clinical trials of fecal transplants in ASD have shown really astounding improvements in social behavior that are very long-lasting. So there's a lot of promise in this. We also know that microglial development depends on the gut microbiome. So germ-free mice have immature and hyper-ramified microglia. And I didn't show you these data in our model. We also see hyper-ramified microglia in our diesel stress males only. And we think that they're immature because, again, they're delayed in sort of losing that heterogeneity compared to controls. And work from Sonia Gorel's lab has shown that male microglia are particularly affected by the gut microbiome early in life. So a lot of things pointing to this. So our first approach with this was simply to sequence the microbiome of the cecum, a part of the gut that holds a lot of the bacteria in mice and in humans, using 16S sequencing. And looking broadly at those metrics here, this is a little small, but I'll walk you through it. First, we looked at the sort of classic alpha and beta diversity metrics, and there are a number of these, but alpha diversity broadly is asking, how diverse is the microbiota within a given individual, and does this differ by group? And indeed, we found in one metric in particular called species evenness that we have actually a lot more species evenness in our 2-hit males earlier in life, but then this dramatically declines such that they have a much lower evenness later. And no changes in females. So again, consistently no changes in females. And then looking at beta diversity, every single beta diversity metric that we looked at was significantly different by group. This is now asking more about whether different groups cluster together or not, or whether they cluster apart. And you can see that in males, very clearly, our 2-hit males cluster distinctly from our controls, whereas females, again, they're all mixed in together. There's no clustering. There's no significant differences in females. We've also begun to really narrow in on what are the actual taxa of bacteria that might be changed in this model. And you can look at this at several levels, of course, like family and genre and species and subspecies, etc. And we're down to the genus level, at least, and seeing that there are 2 major genera of bacteria, Bacteroides and Parabacteroides, which are significantly depleted in our diesel stress males. And we're now doing shotgun metagenomic sequencing to see more clearly what species and then strain level might be changed. And this is really interesting because there are reports in human patients with autism that these same genera are depleted in autism. So we're very interested in whether we may be able to supplement back at this level and restore function. Now, the other thing we've looked at, though, is that, the gut isn't just what's in it, right? But the gut is its own thing. It's its own structure. And so we were very interested in examining the gut itself. And here we found actually some of the most striking changes we've ever seen. So this is work by Dr. Marcy Kingsbury, who was a senior scientist in my lab at Harvard, and now is an assistant professor there and has her own lab. And she's become an expert on the mucosa and examining the structure of the gut, in which you can basically take these sort of coronal sections and look intensively at villi structure and see how the gut is forming. And here she found that, and hopefully you can just see visually the magnitude of this change, that in our diesel stress males, they have a dramatically increased villi length in the ileum. In this case, both males and females, although the effect is larger in males once again. And this is a really striking phenotype because it's actually consistent with many nutrient malnutrition phenotypes. And so we're very interested in what it is that may be initially causing this gut structural change. And of course, you may have also heard about the so-called leaky gut in autism. And we wanted to look at tight junction proteins, which form these sort of junctions between the villi. And this is what allow nutrients to sort of pass in and out. And so you always want some leakiness, but not too much. We examined 3 different tight junction proteins, claudin, occludin, and CO1. And in every one, we see this very striking sex difference once again, where basically male 2 hits, diesel stress go down, and females actually go up. And so it's just a totally different response between males and females in the gut. So to just summarize all of that, what I've shown you so far, of course, we see social behavior changes in males. We see striking gut microbiome changes, the composition of the gut, as well as intestinal epithelial changes, both villi length and tight junction proteins, primarily in males. So the question here was, could we potentially rescue social deficits by manipulating the gut at birth? And in this case, manipulating the gut microbiome, so the contents of the gut. And we know that if you simply cross-foster pups the day of birth, so sort of the second they're born, as quick as you can, you go in and you put them with a foster mother. It turns out that even though you acquire the microbiome through the birthing process itself, and some of you may be familiar with that literature, if you cross-foster very quickly after birth, those pups will attain the microbiome of their foster mother within the first few days, and that will be a very persistent effect. So that's what we did. We simply switched them from their birth mother with the same condition to another birth mother with the same condition, or the opposite condition. And here, the really critical comparison is that if you have a diesel stress born pup and you go to a healthy control mom versus another diesel stress mom, what happens? And so first, what we found is that cross-fostering completely rescued the microbiome changes that you see as a consequence of diesel. So here's that critical group of diesel back to just another diesel mom. It's rescued if you now go to a control mom, both alpha diversity and beta diversity. Beta is a little harder to see here, but this is the diesel here, way out here in red. And if you cross-foster them, they now look much more similar to the vehicle, the control group in blue. And if we look at the behavior, this is just looking at pups from diesel stress moms going to either diesel stress foster mom or control mom. You can see that if they go to a control mom, we can rescue sociability. And this is really driven by this increase in social investigation in this group. So we can rescue the microbiome changes, we can rescue the behavioral changes. Now, the critical question becomes, what are the mechanisms? Importantly, I want to emphasize again, we're always worried about, is this something that we're not thinking about? And in this case, a big one could be, maybe it's just maternal care. So we very carefully looked at maternal care in this model. And what we found was that if anything, the pups that go to the moms that were treated with diesel and stress they're actually caring for their pups more. So they're spending more time on the nest and they're giving more nursing and licking and grooming. So they are not bad moms. They're really good moms. They're spending a lot of time with them. But that's not enough to rescue whatever is happening, we think, prenatally as a consequence of that inflammatory cascade. So what about microglia? We don't know yet. So we're working on this part of the puzzle where it's conceivable that we are rescuing microglial function in those cross-fostered pups, and that's what's then leading to the social behavior rescue. It's also conceivable that it could be an independent and parallel mechanism. And so we're interested in sort of looking at all of these possibilities. With such a big problem like autism, we think the more interventions, probably the better. And so some of the things that we're quite interested in within the gut are what the microbes are producing, like short chain fatty acids and bile acids that could directly enter the brain and impact the way the brain functions, as well as sort of this synaptic connection between the gut and the vagus nerve that's been demonstrated here by our colleague Diego Bojorquez as sort of looking at that mechanism. And so with that, I'm going to end, hopefully on time, and thank the members of my lab that have been involved in all of this work. I have such an amazing group that I'm very, very grateful for. Wonderful collaborators that I highlighted, Koff and Chala at Duke, and then Caroline Smith, who's really been a driver of a lot of the work, including the microbiome work. Karina did all of the synaptic work, all of the microglia work. Marcy working on the gut structural changes. This work was funded by NIEHS as well as the Robert and Donna Landreth Family Foundation. This is how you can find us. I'm happy to answer any questions if we have some time.
Thank you so much, Staci. That was amazing and definitely right on time. So yeah, if people have any question, again, you can write them here in the Ask a Question button. I think we already have one, so I will start from this one. It is from Ritika Sat. I hope I said the name right. What are the controls used for the 2 hit mice, 1 hit mice, or no hit?
So the control for the diesel exhaust particle is basically just a little puff of air, so just a little filtered air as opposed to the particles in the air. But they still get sort of restrained, so we can do the same we sort of puff it down their trachea and it goes into the lung. So they get all the same stress of being handled and all of that sort of exposure. And then there's no control for the stress component. The control is simply normal housing and bedding. So we have that sort of two hit. And in previous experiments, we have determined whether there's an impact of just the stress, the bedding removal itself without any other manipulation. And we've also determined what's the impact of just the particles, the diesel particles on. And the two are required. So you can't really see any impact on social behavior without both combined. So it's a really intriguing synergy. It's not an additive model, it's a synergy because you see nothing with each alone.
Thank you. We have another question from Asia Rolls. Very cool work, Staci. I wonder if in addition to the changes in the gut epithelia, there are also changes in the epithelial cells in the brain borders.
Ooh, that's a great question. Yes, we are interested in that and working on that now. So Danielle Rendina in the lab, who's working on the microbiome differences, is working on the CVO regions to see if there might be changes there. But broadly, it's a great question of whether there's sort of we know, in fact, in autism, that you see changes in tight junction proteins in both the gut and in the border regions of the brain. So yeah, fantastic question. We don't have data yet, but we're working on it.
Okay, another question from Rita Cardoso Figueiredo. Thank you for a very interesting talk. I was wondering if you know if the roles of microglia in the adult, given synaptic pruning happens mostly in prenatal periods, right, are dependent on the microbiome and stress.
So the question is the pruning that occurs in adulthood or later in life?
Yeah, I think it's basically if the roles in microglia in the adult are dependent on the microbiome and stress. So because she's arguing that synaptic pruning happens mostly in prenatal
Well, actually,
periods.
It happens mostly postnatally, in early postnatal periods. There's usually more synapse formation prenatally as well as postnatally. Most of the pruning happens in the first postnatal weeks, although it depends on the brain region. And then other labs, like Beth Stevens' lab and Dory Schaefer's lab, they've shown that you can have a reactivation of pruning in pathology, certainly much later in life. We don't know yet if, for instance, the microbiome manipulation that we did is changing synaptic pruning in microglia. That's what we're working on now. But I guess maybe the larger question is it a persistent enough effect that you would affect it much later, like in adulthood? And that's a great question. I think what we've seen so far is that the phenotype that you have essentially programmed early in life as a consequence of these exposures can be extremely long-lived, so they can persist into much later life.
And this kind of relates a little bit with one of my questions, because is there any possibility of treating these kind of diseases by modifying the immune system in somehow?
So you mean later in life?
Right.
Yeah, so that's a great question. So we've also done a manipulation. I didn't show it just to be on time, but we've done a manipulation at weaning. So there's lots of ways you can modify the microbiome. Of course, the earlier the better. But we have done a co-housing manipulation where we weaned the mice into groups that were the opposite group, essentially. So we would wean a diesel stress exposed mouse into a group of control mice. And after a while, because mice are coprophagic, basically, they acquire each other's microbiomes. And so we were wondering if that could rescue some of the social deficits as well. And it did. but not as well. So it was wasn't as striking of a rescue. So, but there's definitely hope for doing it later in life. Now that was through a direct manipulation of the microbiome in terms of that co-housing. But your question was more the immune system. So one of the things we're very interested in right now is whether we could use short chain fatty acids, which are produced as a consequence of certain bacteria. And in fact, they're produced by the bacteria that are depleted in our two hit mice. And so we think that maybe they're not producing enough of these short chain fatty acids, which are important for all kinds of things, but including for microglial function and homeostasis. And so we're interested in whether we could supplement back and then rescue behavior, rescue deficits that way. So it's a great question, and that's the most relevant for patients, of course. Is it too late, right? No, we don't think it's too late, but we think we just need to be able to, narrow in more on these potential treatments.
Yeah, that's so interesting. And also, I think I can ask maybe the last question, because at the beginning, for me, it was kind of intuitive of thinking, okay, if the mother is in an environment that is not good and it's causing her stress, that at the end, that will make the pups kind of feel, okay, this is bad. I need to maybe either stop socializing or things like that. So if there's anything related to the immune system, to start maybe stop the transmission of any kind of virus or bacteria or something. I mean, we can hopefully not, but maybe we can relate it to what it's happening nowadays. I don't know if people are already starting studying autism in monks that when they are in the age of coronavirus.
Yeah.
But one of the things that was kind of puzzling for me is that I will also expect that the pups are going to also change the object interaction, because they don't know why it has to be social. They don't really know what is bad in the environment. They just know there's something bad, and then why is it a change just in the social interaction and not with any other thing in the world?
I mean, it's an excellent question, and it's one thing that we've been grappling with for, I think, as a field, right? And that is what are these assays sort of capturing, and how does that relate to what you see in patients, right? Because, and one thing I didn't show you, which was again, in the interest of time, but it will be in the paper when it's all in revision at the moment, that early part of the story, at least, it will be in that paper, which is that when we did the multi-unit recording with these mice, where we did 10 repeated social interactions so that we could collect enough data to do this massive social circuit analysis. What was really, really interesting, and these were adult mice at this point, so not juveniles. We saw a social deficit in juveniles, where they're not preferring the object. But when we do that repeated social task, our two hit males are actually more social by the end, because what happens is the control mice start to habituate. So they start out super social and then they sort of decline, which just happens with normal interactions. We're like, well, I already know you. I don't need to ask you 10 questions about your life right now. And our mice, they never habituated. So it's really interesting phenotype, because it's actually sort of consistent with many patients with autism that have not, it's not that they're not interested in social interactions, they're just sort of different, right? It's that sort of, their social interactions are different, and we seem to capture that with that assay. So it's really fascinating. And of course, we never want to try and say, oh, this is just like people, but it's interesting, it's intriguing how many sort of corollaries there seem to be. So I don't know if that was really what you're getting at, but I think social behavior adapts and it changes according to the environment, and that's a really important consideration. And in each case, we sort of see a difference in our mice. So
Yeah, I agree with Rita. She's writing that it's so fascinating, and I completely agree. So yeah, I don't think we have any more questions. So thank you so much for your talk, and thank you everyone for joining us today. Our next speaker will be Soo-Jung Park next Monday, who will talk about nutrition, metabolism, and decision making in humans. So I hope to see you all next week. Bye.
Thank you so much.
Thank you. Thank you.